Good morning, ladies and gentlemen, farmers, colleagues all. It is indeed a pleasure for us to be together here today to discuss some new strategies for management of major rice disease in Guyana. It is our first webinar in a series of presentation to mark, to celebrate Agriculture Month in Guyana. And the theme for Agriculture Month in Guyana, I must remind you, it's invest in agriculture for poverty reduction and sustainable development. Dr. Rajendra Prasad, AA, a colleague of mine, pathologist at the Rice Research Station, has just completed his PhD studies and have been heading the plant pathology department for over a number of years. So today, he and his colleague, Ms. Donata Magan, scientist, just back from studies in India a year and a half ago, will share the presentation. The mandate of the research station is to develop new technology for the farmers. We have committed over the past years. In so doing, we have delivered many technologies, and particularly in varietal improvement, disease management techniques. We have developed in seed production, in strategies to combat paddy bug, and soil nutrition amendments, and general agronomic practice. In many areas, you will have further down the month, you will have also Dr. Gansham Payman will be discussing some aspects of red rice management in Guyana. Also, we'll have Dr. Vivian Baharali looking at paddy bug management strategies in Guyana. Also, our head of extension unit, Mr. Bisisar Prasad, would also highlight the improved agronomic practices has been approved by GRDB and was designed by the farmers, the scientists, the extension officers. In fact, is the best practice for rice in Guyana. And myself at the end would also discuss some of the findings later in the month of the new candidate, the variety that we are testing for possible release for cultivation in Guyana. Before I will go straight ahead to introduce you to Ms. McGowan to do the first half of our presentation, titled New Strategy for Management of Rice Disease in Guyana, and then Dr. Rajendra will follow through. Over.
Thank you, Dr. Prasad. Good morning to everyone joining us. As mentioned, our presentation is entitled New Strategies for the Management of Major Rice Diseases in Guyana. Today, I'll be focusing, Today I'll be focusing on the major rice disease in Guyana, conditions favoring these diseases development, the economic importance and symptoms of these major diseases. Rice is considered to be one of the most important food crops in the world, as it is relied upon as a staple food by billions of people worldwide. A crop loss for any reason can pose a threat to starvation. In Guyana, The rice industry is one of the most important agricultural industries, uh, contributing approximately 10% to the GDP. Only recently, we've had reports that in excess of $30 billion was generated through rice exports between the month of January and August. And by the end of 2020, as much as 1.1 million tons of paddy, will be produced. Rice is also one of the largest users of agricultural lands, with approximately 95,000 hectares being double cropped annually. Now, this may all be well and good for Guyana, but looking at it on a larger scale, the world population is set to increase by 2050 to 9 billion. This all indicates that we will have a larger population to feed on smaller land, as much of the arable lands may be converted for industrial purposes. Not only that, but climate change and global warming may see a rise in sea levels. So many of our arable lands may be covered. So in this situation, we will want to make every grain that is planted count because we have more mouths to feed and less land from which to produce food. Now, one of the major factors contributing to rice disease, to rice loss, is diseases. As much as 14% loss in rice production is due to fungal diseases. In Guyana, the major rice diseases are blast, brown spot, sheet blight, sheet rot, and grain discoloration, while there are a few minor and new emerging diseases. Now, these new and minor diseases do not cause significant economic losses, but however, as I mentioned, due to climate change, the conditions favorable for the proliferation and spread of these diseases might see them being elevated into major diseases. One of the most crucial diseases to rice production is rice blast. This disease can cause from 5 to 30% in moderate cases. But when the conditions are favorable, we can see a total crop loss. That means 100% crop loss can occur. It can affect all above ground parts and can affect all growth stages. Now, the reason this disease is so difficult to control is due to the pathogen's ability to survive harsh conditions and spread easily from field to field. Not only that, but varietal resistance may also decline or break down since the pathogen may develop new strains and become more virulent. Some of the conditions favoring this disease development and spread: low soil moisture, prolonged period of rain, cool daytime temperature. In anywhere where there is prolonged dew formation on the leaves, this can increase the spread and the proliferation of the disease. Symptoms on the leaf may vary depending on environmental conditions, the age of the plant, and the resistance. However, initial lesions or the affected area appears gray-green, water-soaked, and with dark green borders, whereas older lesions appear spindle-shaped with whitish to gray center and necrotic borders. In susceptible cultivars, these lesions may expand, they may coalesce, that meaning they will join together and take maybe several centimeters, they may spread over the entire leaf, killing the leaf. Whereas in resistant cultivars, the lesions remain small and brown to dark brown. When it comes to the color of the rice plant, this is the area at which the leaf joins to the sheath of the stem. In this area, you will see necrosis at the area joining the tooth tissue, or darkened area to indicate that that portion has been killed. So you will see necrosis in this area. This disease can spread from that area up to through the entire leaf, killing the entire leaf, and may extend a little bit below the onto the stem. Whereas in the case of the neck blast, the neck is the portion of the stem that extends above the leaves and to which the panicle is attached. Now, at this point, you may find a dark brown to blackish lesion, and unfilled grains may be produced, and the panicle of itself may fall over. Symptoms on the panicle: we will find lesions on the branches and spikelets. The branches may break at the lesions, and there may be empty grains in the lower portion of the panicle. Whereas on the seed, brown spots or blotches may be found. Brown spot. This is another fungal disease which affects the rice production. This is a very important and also crucial disease that could result in yield loss from 12% in moderate cases to 45% in severe cases. This disease is said to be one of the main contributors to the Bengal famine in the 1940s, resulting in as much as 2 million deaths, since several rice crops would have been lost and resulted in starvation in Bengal. The disease can affect the coleoptile, leaves, leaf sheath, panicles, and glooms. It can occur at all stages. However, it is more evident during the tillering to ripening stage of the plant. Some of the conditions favoring this disease: high temperature, high humidity, unflooded and nutrient deficient soils, or sometimes soils which accumulate toxic substances, and the leaf would have to be wet for approximately 8 to 24 hours. And some of the major sources, now these sources are similar for most of the diseases that I will be discussing. This would be the infected seeds, volunteer rice, infected rice debris, and weeds. So as long as these sources are in your field, it's possible that the disease can be present on them and can later transfer to your crop. Some of the symptoms of the brown spot disease: at the seedling stage, small circular yellow-brown lesions may girdle the coleoptile. Whereas at the tillering stage, the lesions are initially small, circular, and dark brown to purple. While developed lesions are circular, oval with light brown to gray center, and bordered by a reddish-brown margin, and it may have a yellow halo surrounding the entire lesion. In susceptible varieties, the lesions can be from 5 to 14 millimeters long. The leaves may then wilt and die, whereas in resistant varieties, the lesions are brown pinhead in size. Lesions on leaf sheath are similar to those on leaves. Infected glooms and panicles have dark brown to oval spots, and infection on florets leads to incomplete grain filling and reduction in grain quality. Now, this fungus can penetrate grains and cause what we call pecky rice. This is just another name for discolored grains. In certain varieties, this disease may be confused with blast disease. However, just to confirm, the brown spot disease is more circular with a grayish center, a dark brown margin, whereas the blast lesions would be sort of a spindle or a diamond shape with gray center. Another major rice disease caused by fungus is sheet blight. It affects leaves and young tillers. Yield loss can be from 20 to 50% in susceptible cultivars. Plants are more vulnerable to this disease during the rainy season. And also, some of the conditions are listed here. The symptoms of this disease is an oval to ellipsoidal gray-greenish lesion on the leaf sheet. This lesion begins at the uh, water level or the soil level. Now, in favorable conditions, these lesions can expand and get onto the leaves, at which time it appears irregular with a gray-white center and brown margins. So after it gets onto the leaf, that's when it has the possibility of spreading to other tillers, other plants, and can spread throughout the field. Sheet rot is a fungal disease which is most destructive just before the emergence of panicle. Some of the conditions favoring the disease is increasing plant density and high nitrogen fertilizer, high temperature and relative humidity. Also, it is important to note that uh, any opening that may have been created through a wound or injury to the plant, let's say it was attacked by stem borer during panicle initiation. This opening can cause the fungus to enter into the plant and make it and affect it. The lesions begin on the uppermost leaf sheet, that is enclosing the young panicle. Lesions are oblong and irregular spots with reddish margins and gray center or brownish throughout. The panicles remain partially emerged, and panicles that have not emerged rot, and the florets turn red brown to dark brown. Uh, if you look closely, you may see a powdery white substance on the outer leaf sheet. This is some portion of the fungus that will remain there that can cause also spread the disease. And in some cases, there is confusion between this disease, the sheet rot, and the sheet blight disease. However, it is important to note that the sheet rot, it only affects uh, it mainly affects the plant during the panicle initiation stage, as before the panicle emerges, and it prevents the emergence of the panicle. However, the sheet blight disease does not interfere with panicle emergence, and it begins lower on the plant, whereas the sheet rot disease is much higher. The final major disease that I'll be dealing with here is the grain discoloration disease. Now, this disease was observed to be caused by many fungal organisms, but in Guyana, the main organism has been found to be the Curvularia species. Uh, the discolored or affected greens are often referred to as dirty greens or black tip. The symptoms appear as small dark imperfections on the dorsal surface of the rice kernels and has a lesion-like appearance. Due to the etiology of this disease, it is unlikely to be detected in the field before harvest. Since you have to remove the hull of the grain, that is the only time that you would notice this imperfection on the grain. So it's very difficult to determine if the disease is present in the field. Just an estimated cost analysis here. Now, if you're a farmer and you're averaging 40 bags an acre, and you are affected by one of the diseases that I've mentioned, let's say at a 12% infection rate, you're looking at a loss of approximately 8 bags per acre. And should be making 3 $1,000 per bag, that is loss of $24,000 per acre. So this is just to give you an estimate on the losses that can occur if disease isn't managed within your field properly. So I'll now be handing you over to back to the chair to Dr. Minder Prasad, who will continue to chair this meeting. Thank you.
Thank you very much, Donata, for guide us through the identification of the different disease, which is so important, and it's actually crucial for application of any management strategy. The first thing you need to identify the disease, and there's always confusion in disease, as you have highlighted in sheet rot and sheet blight. Also, importantly, there's a confusion between at the farmer level in terms of the brown spot and rice blast. And you have also highlighted those two lesion types that are very different. The blast has been the eye-shaped lesion, whereas the brown spot is more of a circular type of lesion. So now I'll ask Dr. Rajendra to continue on the same path to look at the strategies and some of the work that he has done in the recent past and some of the strategies that he advising the farmers to use to combat the various diseases that are prevailing in Guyana. Over to you, Dr. Raj.
Good morning, and thank you very much, Dr. Prasad. So I'll continue. With the presentation. So what was done before and where we are presently, in terms of what work was done before. No, the screen is changing, the PowerPoint. Right, sorry about that. What was done before? Generally, the farmers was heavily relying on the use of chemical as one of the major means of controlling disease. And more so, they were using the chemicals that were old and outdated, right? So they were heavily relying on chemicals at the management level, as well as no research work was done prior to the 1980s in terms of the resistant screening to identify lines or germplasm that possesses resistant to the major diseases. And there's no available information or proper document information prior to the 2000 in Guyana in terms of the use of alternative management approach for the management of these diseases, as well as no scientific study in terms of new generation type of fungicides. So in terms of that, the research was prepared and developed with these in mind. And we have three areas of focus, which I'll discuss as we move along, is to identify blast and sheet blight disease resistant genotype from the advanced rice germplasm, to evaluate the efficacy of some plant extract and bioagent along with new generation fungicide against these two diseases, as well as to identify the causative agent and to develop strategies for the management of the grain discoloration pathogen. Now, let's first look at the aspect of the blast screening work. In terms of the screening work, when we say hotspot location, we mean that those locations are ideal or conducive for disease development, particularly the rice blast. So those environmental condition, environment or area was selected. We choose Anfawak, Black Bush, and Kanji as three of our hotspot location based on previous research work. And this research is carried out basically in the two rice growing cropping season. The first, which has been the spring crop, starting November, December, and end March, April, and the second being the autumn crop, May, June, and end September, October. Um, in terms of our seed source, the GRDB has in its germplasm over 5,000 plus rice germplasm accession from various parts of the world, such as flour, Cardi, Erie, Brazil, Suriname, and some that we have developed from our own local hybridization program or from our local crosses. Um, However, for this particular study, we have selected some of the cream of the material uh, taken from the observation yield trial, advanced yield trial, and some of our varieties that is presently in the farmer field, along with one of the susceptible check, which is the rustic variety. The screening work was done using what we term an upland blast nursery um, in the different areas. And the scoring was done using 0 to 9 scale. Uh, um, when the susceptible check, which is a rustic, reach a scale, as you could see in the image below, when the susceptible check reach a stage of 8 to 9 minutes completely dead, or then you actually go in to do your final scoring and selection, right? In terms of just to give you an idea, what the standard we use is the SES standard with a 0 to 9 scale. So when we say 0 and then we look at the host respondent, so the host response when it is 0, when that means it has no lesion observed, and it's meaning that plant or that line would be highly resistant. Likewise, when the score is 1 with a small brown speck of a pinpoint size, that means that particular line would have been resistant. And likewise, we move on 2 and 3 based on the lesions that we observe. Those are given the host response. Where we are and where we was and where we are now in terms of our resistance screening program. In terms of our variety, at release, all of our variety were released with highly resistant or some moderate level of resistance. But over time of cultivation, we have seen some of the resistance has been broken down. And as my colleague before explained, because of the nature of the blast pathogen, it is capable of changing its form, forming new race, new strains, and so on, and overcome those resistant genes. So what you have seen, for example, in the GRDB13, it was released as a moderate resistant line, and it now came down to a highly susceptible line. Likewise, we have seen BR444 and 196 and G98 135. Those has moved from being resistant status to being highly resistant, and so on. So that's why our resistance screening program has to be continuous. Slide. Yes. Slide. Right. In terms of looking at the blast resistant data coming out of our screening trial, we found because since this material that we have used, majority of the genotype came through from the these are the cream of our material from the observation yield trial, advanced yield trial. So they were bred with some level of resistance. So we have seen from this data, majority of the lines have shown highly resistant or resistant status. And, and as you can see in this table here, one of our candidate line that is now we're in farmer field in the on farm trial has we have observed that line to have from since in the 2015 research, we have seen that line to possess high resistant to resistant status. Apart from that line, we have also found some other line having similar resistant status, along with our some of the popular variety like the GRDB10, GRDB9, and so on. Right? In terms of the response of these genotype with the AMI analysis, which looks at the interaction of these lines or these genotype with the environment, we have seen that there's not much, the resistant genotype cluster towards the origin, which shows that there was not much influence in terms of the environmental condition in terms of resistant with this genotype. As you see, the resistant cluster towards one end, while the highly susceptible line, which is rustic 103 in this case, on the other side. Now, in terms of concluding and giving a recommendation based on this study, 11 of those germplasm lines that were found to be highly resistant and resistant, we are now recommending them to be part of the hybridization or the breeding program, or being selected and move forward for further testing work, like how we had the GR, the B12, 259 now is already advanced into the farmer's field as in the on farm trial, and would be after some time would be considered as a new variety. Along with those entry, we have found some of our susceptible, some of our popular variety, like the GRDB 10 and 9 and so on, to be high, to maintain their resistance status. So what we are recommending to the farmers is to use those variety that has high level of resistance in area that is considered as hotspot or have condition that favor the blast disease development. In terms of this particular experiment, it found that the ideal test environment was the Kanji location, and the susceptible check has recorded the highest blast score, which has given the reliability to this particular study. In terms of the sheet blight screening, this is basically done under artificial and local and artificial inoculated condition as well as field condition. And these are some of the data that we record in terms of that particular screening exercise. Like we recorded the percent disease severity, the area under the disease progress curve is determined in order to identify variety that is have a slow blighting effect, the apparent rate of disease development progress between the observation also is being calculated along with the AMI analysis. Now, from these 101 genotype that was evaluated, we found a large percentage to show immune to resistant reaction, and those is highlighted below in this line graph. While on the other side, we found 2 to have high level of susceptibility along with our check line. When we did our AMI analysis, we found that there were some level of interaction effect which influence the resistant and susceptibility of these genotype, because under the different environmental condition, we have seen some level of variation, right? So in concluding, we have found 14 genotype showed resistance status range from immune to resistant. And within this as well, we found some of our popular variety like the G98135, GRDB12, and BR444 also form part of that 14. So in recommending now to farmers, if you know that this area within your cultivation is prone to sheet blight disease, right, which is another major disease for us, we are recommending that you use this variety that has that level of resistance status in them. And we also recommending that these lines be further be evaluated and studied for uh, by the breeder and the research team so that we can see what has contribute to the level of resistance in these material. In terms of these line as well, 13 of these 14 showed slow sheet blight development with a relative low area under degrees progress score, and 11 show reduction in apparent rate of disease development. So I already mentioned the environment influence had some uh, effect, while the best environment we found for this particular study was the one at Anfawagbak, and it was during the autumn crop. It was closest to the average and ideal test environment, and we found two of our genotype. Um, One which is our susceptible Czech variety rustic, along with a new other genotype. In terms of the second research area in focus is to evaluate the efficacy of plant extract, bioagent, and new generation pesticide against the blast disease on the laboratory and field condition. In the laboratory, we have evaluated 11 different plant extract along with a control or a check. And out of these 11 extract, we found uh, five of these extract to give more than 80% reduction in the growth of that particular fungus. Mind you, all these diseases that we are talking about so far are caused by fungal microorganism. So, and this study is done on the lab under the controlled environmental condition. Similarly, we did this the screening of three bioagent against this particular pathogen, the Azobacter SAG19, Bacillus OG2L, and Bacillus subtilis OG2A. And what we have found, all these three bioagent had shown more than 50% reduction in the growth of the fungal microorganism under the laboratory condition. Now, from that, we have selected only the best performing products and take to the field level for evaluation. And it was done on the two season, the spring and the autumn crop. And what we have found from this here, we have found the black stage, bale, and madar extract at the 10, 15, and 5% respectively, along with the two of the bioagent and two of the fungicide to give the best result, which is the Antracal and the Nativo, along with the Cyrenade and the Czech fungicide, the Fuji one. They all showed significantly better result as compared to the control treatment. And you can see that from the statistics. Now, in terms of the percentage reduction, increase or decrease in green weight, we have over the control treatment, we have seen for these better performing treatment, we have seen a more than 15% increase. As you see, we have highlighted in the green in this particular graph, the above 15% increase in these better selected treatment. So in concluding here again and recommending, we know that these three products as being the black stage, the bale, and the madar has shown high level of efficacy along with the bioagent. So we are recommending this as a first line of approach for farmers to use against the particular pathogen in the event there's a outbreak or there's a need to apply control measures. Because if you use the highly resistant cultivar and so on, you will not need this. And those two, the bioagent and the plant extract, once they are available, they are more environmentally friendly. For example, the plant extract is available in abundance, and it can be a cheaper means of control as well. And if that is not available, then you can go with the use of the recommended new generation type of fungicide rather than going with the old type of fungicide. And we can again see here a note with these fungicide and recommended products not only give a control of the pathogen but also shown a positive influence in terms of yield. Um, in terms of the sheet blight management, we are looking at a similar set of plant extract as well as the bioagent against this particular pathogen, the Rhizoctonia solani, under laboratory condition first. And we have found four of this plant extract: the lemongrass, the thick leaf thyme, the onion, the marigold, and the clove showed significant reduction, which is more than 90% reduction in the growth or slowing down the growth of the fungus, which we term percentage inhibition of mycelial growth, right? Likewise, we found out of the three bioagent, we found one to be effective, which is the Bacillus cereus OG2L. Now, again, we did the similar as before. We combined those better performing products and evaluated with the chemicals, and we found the lemongrass, the thick leaf thyme, along with the Bacillus OG2L strain at the 2 grams per liter, along with the, the Anthracal, Nativo, and Serenade, give the best result over the two test period, which is also comparable with our standard Fuji-1 check. And this treatment has shown significant reduction in the incidence of the sheet blight disease when you compare them to the control. Now, just we showed this graphically, the percent increase, percent decrease in the disease severity over the control treatment, and we see the better performing treatment has shown greater than 70% reduction in the disease incidence. And mind you, again, we have seen similar the result where there is more than about from 8 to about 36% increase in the grain weight. So, in concluding again, for this particular trial, we found the two plant extract, the lemongrass, thyme, and at 15% concentration, along with one of the bioagent, Bacillus cereus, OG2L, and the three new generation type of fungicide, the Anthracal, Nativo, and Serenade, to be the most effective means of controlling this pathogen, and is recommended to be used by the farmers. Now, let's turn our focus to the third area, which is to look at the grain discoloration. In terms of the work, we basically did isolation from the pathogen across the different rice growing region, following the blotter tests agar plate and the ISTA protocol. The pathogenicity test was also carried out using following Koch's postulate. Um, and the in vitro screening was done in the lab following the similar technique which we use is we call it the food poison technique. Basically in this technique where what we do is poison the food, which is in this case is the PDA with the chemical, and then we plate it and put the fungus and do our evaluation in the lab under that condition. While the RCB design was utilized for the field, and the finding from that field trial again was taken to the farmers' field as demonstration and recommendation to the farmers. In terms of the pathogen associated with grain, we found as my colleague mentioned before, the Colvularia species to be the most predominant microorganism. However, from our studies in Guyana, we have also found Bipolaris oryzae, Sarcrodem oryzae, Alternaria, and Aspergillus, along with Fusarium on the grain. And this was further confirmed by CABI in the UK. They have confirmed that the pathogen that predominates on the grain with the discoloration was called Valeria lunata. And this was done up to a molecular level where the IT region of the rDNA reveal 100% match to Valeria lunata. And you can see those are some micro images taken from the under the microscope, showing the spores as well as the conidia. The conidiophore is basically the mycelium structure that you saw in the picture below. Now, in terms of the laboratory screening or the in vitro screening, we have evaluated these fungicide at different rate, and we have found all to show significant reduction in terms of the growth of that particular, the Corvularia lunata under the control laboratory condition. So we select the best and with the lowest, the lower rates, and evaluate them under field condition. And this was done during the spring crop 2019. And what we have found from that trial, we found a range of 33 to about over 60% reduction in incidence of grain discoloration over our control. And then we look at the aspect of the grain yield and so on. And likewise, we have seen an increase, a slight increase in terms of the grain yield when that was done. And this was repeated during the autumn crop of 2017, and the finding was almost similar, right? So from that, those two trials, we took the better performing products, and which was the Amistar Extra, the Glory, Antracal, and Carbendazim, and then we take that out to the farmer's field in more demonstration type of exercise. And we have found a range of from 48% to 53% reduction in the incidence of the grain discoloration over the control treatment in this particular case. And likewise, it was done throughout all the rice growing regions, and the data shows the similar kind of finding where you had the cases, most of the cases we found is reduction, not a 100% reduction in incidence of grain discoloration, but a significant percent, which is greater than 50, as well as a increase in the grain yield, which is above 20%. So in concluding, the prominent microorganism associated with the grain discoloration is the Corvularia lunata. And as I mentioned, this was confirmed by CABI Those are the chemical listed here that shows that was evaluated under laboratory condition and showed very good or promising results. So we took them to the field trial, and from the field trial, we found these chemical, the Amistar Extra at 1.5 ml per liter, the Glory at 3, and the 5 grams per liter, along with the Antracal at 5 grams per liter and the carbendazine at 1.5 ml to give excellent control, which shows more than 50% reduction in the incident. And the similar was repeated in the regional trial, where we found a range between 48 to 58, 48.58 to 92.85% reduction in grain discoloration incident when we apply the treatment. These particular chemical, as I mentioned, and not only the reduction in incident we have observed, but also a 12.28 to a 55.18 increase in yield was observed over the control treatment. So let's summarize in a general way how we can go about manage these disease, because I spoke about individual and so on. So in terms of the general control strategy, we are recommending as your first line to use blast resistant and or highly tolerant rice variety or that is available for this particular pathogen. As my colleague mentioned before, resistant variety will not remain resistant indefinitely, and most of these variety may break down after 3 to 5 year or 6 to 8 season of cropping. So our resistant program has to be and must be a continuous one. In terms of our recommended cultural practice for the control of these major disease, we are recommending to sow within the recommended cropping period. As I mentioned earlier, what are the two cropping period recommended for sowing? Practice proper field sanitation. Do not have excessive do not use excessive high seed rate, because that would create a micro environment or a condition within the canopy of the plant that favors disease development. So you need to have your proper plant stands as well. Avoid application of excess amount of nitrogen, and keep the soil nutritional status balanced. So that's advisable to do your soil tests frequently, know your nutritional status, and ensure that you apply a balanced nutrient to your soil, because one from this soil testing result that we have done previously, it have shown some of the elements like the K and calcium, magnesium, and so on are missing. And when these, for example, when you have a area where the K is missing, you'll find an increase in the incidence of the brown spot. So because the plant is more vulnerable in those cases. Keep your field flood at the correct depth, and we're recommending it within 4 to 6 inches, and keep alternate blast hosts under control. Likewise, other hosts. And in terms of the alternative and chemical control approach, the second, which we are using, recommending now, is the use of the plant extract and the bioagent. And these are the plant extract we are recommending and bioagent, and they are effective against, for example, the black stage, bale, and madar are highly effective against the blast pathogen, while the lemongrass and thick leaf at those concentration are effective against the sheet blight, and those two bioagent are effective against the blast and sheet blight, and those are the recommended chemicals and their appropriate rate at the and in the pathogen that they're effective. However, I want to point out, whenever you're applying disease control using these method, plant extract, bioagent, fungicide, we're recommending that you use at least 2 motor blower of spray solution um, per acre. These are some of the other reading material you can refer to for some of these research work that was done and published, along with some other material that we have used. And I must thank you and thank my co-author. And I now hand you back to Chair.
Thank you very much, Dr. Raj and Dhanata McGowan, for a very in-depth presentation, I must say. We heard Dr. Raj emphasize a lot of some of the work. I'm getting a terrible feedback here. Thanks. Yes. In screening, many for diseases, resistant screening, and also we have focused, well, overall you could see that we have been focusing our management strategy. The first line of defense is to use resistant gene. And simply, it is more effective and it is cheaper whenever it is possible. So we go for that. We also see focuses be made now on use of biofungicide. That would be our direction in the future as much as possible. We'll try to advance our studies into those areas to use biofungicide and generally biopesticide for the other pests of rice too. Cultural approach is indeed an integral part of the entire integrated disease management for rice. And the way we manage our fields and the plant itself in terms of the practices that we are conducting in the field to ensure that the plant is healthy. And I want to also highlight the importance of soil health. Dr. Raj did highlight the importance of balanced nutrition to ensure that the plant stays healthy and could use its natural mechanism to defend and to most of these diseases. So soil health is important. And then the last resort, when we go for the more synthetic fungicide. So we will now give the audience a chance to ask questions. You may do so via raising your hands or by texting, and the IT will assist me here. Go ahead.
Thank you. Good morning. I noticed there's a person by the name of Farmer. You sent a text, but if you'd like, you can ask your question verbally also. You can go ahead and do that. Okay, I will read out your question and Dr. Rajendra can respond. I would like to comment on the slide that showed the impact diseases have on production and losses. This type of information. Well, it's not he's not asking a question, he's making a comment. Let me read the comment. I would like to comment on the slide that showed the impact diseases have on production and the losses in dollars to the farmers if not controlled. This type of information should be used by the extension team to educate farmers when they have meetings on the importance of controlling diseases. Thank you for your comment.
Thank you very much.
Thank you very much. much for your comment. Um, I'm sure the extension staff and manager is on board with us, and they would surely take this information and to the farmers level. But I must thank you for your comment.
Any other questions, comments? Well, if we don't have any other question and comments, we'll have to take a leave from this part of our efforts in a series of presentation. And we'd like to thank everyone very much for being part of this presentation. And for those who have sent their messages and comments, I thank you very much. On behalf of Team GRDB, um, we're we thank you very much on behalf of Team GRDB for your participation, and kindly look forward for other presentation in the series on the next one is on the 21st of October, paddy bog management by Dr. Baharali. On the 23rd of October, improve agronomic practice by Mr. Bisisar Prasad, the extension manager. And on the 26th of October, the economic importance of red rice. Dr. Payman, our senior scientist there also, agronomist, will be presenting to us. And finally, to cream this off at the end of the month, myself will be discussing some findings of the candidate variety on the 28th of October. Kindly also follow us on Facebook and YouTube for all the other presentation and also this presentation. And you can keep sending your comments. Thank you very much.